This video will cover everything you need to know for topic 1 of the Edexcel GCSE Biology, which is the key concepts. 1.1a looks at subcellular structures in animal cells. The nucleus contains genetic material that controls the activity of the cell. Genetic material is arranged into chromosomes. The cell membrane holds the cell together and controls what comes in and out of the cell. Mitochondria are where most of the reactions for aerobic respiration take place. Respiration transfers energy that the cell needs to work. Ribosomes are involved in the synthesis of proteins and the translation of genetic material. 1.1b looks at subcellular structures in plant cells. These include all of the ones previously mentioned and the cell wall, which is rigid because it's made from cellulose, and it supports and strengthens the cell. The chloroplasts are the site of photosynthesis, which makes food for this plant. They contain a green substance called chlorophyll. The vacuole contains a cell sap, which is a weak solution of glucose and dissolved salts. It maintains the internal pressure to support the cell. (1.1c) looks at subcellular structures in bacterial cells. The chromosomal DNA means that the DNA is in one long circular chromosome. It floats free in the cytoplasm, not the nucleus. It controls the cell's activities and replication. The plasmid DNA are small loops of extra DNA that aren't part of the chromosome. Plasmids contain genes for characteristics such as drug resistance, which can be passed between bacteria. The flagella are long hair-like structures that rotate to make the cell move. It can be used to move the bacterium away from harmful substances like toxins or towards beneficial things such as nutrients and oxygen. (1.2a) looks at the specializations of sperm cells. Sperm cells contain a haploid nucleus, meaning it only contains half of the chromosomes required. The other half are carried by the egg cell. The acrosome at the front of the head stores enzymes needed to digest through the membrane of the egg cell. It has lots of mitochondria in the middle section to provide energy from respiration needed to swim to the egg cell. And it also has a long tail so that it can swim to the egg cell. The 1.2b looks at the specialisation of egg cells. The egg cell contains nutrients in the cytoplasm to feed the embryo. It contains a haploid nucleus, meaning it only contains half of the chromosomes required. The other half are carried by the sperm cell. Straight after fertilisation, its membrane changes structure to prevent any more sperm entering. This ensures the offspring ends up with the right number of chromosomes. 1.2c looks at the specialisation of ciliated epithelial cells. Epithelial cells line the surfaces of organs. The cilia, which are hair-like structures, beat to move substances in one direction along the surface of the tissue. In the airways, they help move mucus towards the throat, so it gets swallowed and doesn't reach the lungs. (1.3) looks at microscopy. Microscopes use lenses to make things bigger. They also increase the resolution of an image. Resolution means how well a microscope distinguishes between 2 points that are close together. A higher resolution means that the image can be seen clearly and in more detail. Light microscopes allow us to see things like nuclei and chloroplasts and can be used to study living cells. Electron microscopes have a higher magnification and resolution than light microscopes, but cannot be used to study living cells. This means we can see smaller things like the internal structure of mitochondria and chloroplasts with electron microscopes, which allows for a better understanding of how cells work and the role of subcellular structures, which are structures inside the cell, such as mitochondria and the nucleus. (1.4) looks at estimation. To estimate, round the numbers to one significant figure before performing the calculation. Estimation should be used if the question specifically asks for an estimate, but it can also be used to see if your answer is roughly correct. 1.5 looks at units of measurement. To go from a larger unit to the next smaller one, i.e. from millimetres to micrometres or nanometres to picometres, you will need to multiply by 1,000. To go from a smaller unit to the next largest unit, e.g. the micrometre to the millimetre or the picometre to the nanometre, you will need to divide by 1,000. Sometimes it's easiest to use standard form when dealing with numbers, e.g. 0.017 is 1.7 times 10 to the minus 2. Millimetres are times 10 to the minus 3 metres. Micrometres are times 10 to the minus 6 metres. Nanometres are times 10 to the minus 9 metres. And picometres are times 10 to the minus 12 metres. 1.6 looks at the microscopy core practical. The specimen needs to let light through, so it needs to be a very thin slice. A drop of water is added using a pipette onto a clean slide, which will secure the specimen in place. The specimen is placed using tweezers onto the slide. If the specimen is transparent or colourless, then a drop of stain is added, which makes certain structures easier to see. For example, methylene blue stains DNA. Holding it at an angle with a mounted needle, a cover slip is placed at one end of the specimen before being carefully lowered onto the slide. The cover slip is pressed down gently so that no air bubbles are trapped under it. The slide is clipped onto the stage and the lowest powered objective lens is selected. The coarse adjustment knob is used to move the stage up so that the slide is just underneath the objective lens. Then, looking down the lens, move the stage down until it is nearly in focus. The fine adjustment knob is used to adjust the focus until the image is clear. If a greater magnification is needed, swap to a higher powered objective lens and refocus. The total magnification of the image will be the magnification on the eyepiece lens, which is usually around times 10, but make sure to read the question carefully, multiplied by the magnification of the objective lens. You can also work out the magnification if you know the actual size of the specimen and the size of the image. Magnification will equal the image size divided by the actual size. However, the measurements must have the same units. (1.7) looks at enzymes. Chemical reactions typically involve things either being split apart or joined together. The substrate is the molecule changed in the reaction. Enzymes are biological catalysts that speed up the rate of reaction in useful chemical reactions without being used up or changed in the reaction. Every enzyme has an active site where it joins to the substrate to catalyse the reaction, forming an enzyme substrate complex, or an ESC. Enzymes usually only work with one substrate, so they are said to be specific. For an enzyme to work, the substrate needs to fit in the active site. This means that if the substrate doesn't fit, then the reaction will not be catalysed by that enzyme. This is called the lock and key mechanism, because the substrate fits into the enzyme like how a key fits into a lock. 1.8 looks at the denaturing of enzymes. If the temperature is too hot, some of the bonds holding the enzyme together are broken, so the shape of the active site is changed. This means that the substrate no longer fits into the enzyme, and the reaction is no longer catalysed by that enzyme. The enzyme is not dead, because it was never living in the first place, but it is instead classed as denatured. If the pH is too high or too low, then it interferes with the bonds of the enzyme. This again changes the shape of the active site, so the substrate no longer fits into the enzyme, and the reaction is no longer catalysed by that enzyme. This again denatures the enzyme. 1.9 looks at factors affecting enzyme activity. There are three main factors that can affect enzyme activity: temperature, pH, and substrate concentration. Increasing the temperature at first will increase the rate of reaction. However, if the temperature is too hot, it will denature. All enzymes have an optimum temperature where they work best at. In humans, this is around 37 degrees C. All enzymes also have an optimum pH that they work best at. Typically, this is around neutral, pH 7, but not always, as some enzymes work best in more acidic or more alkaline conditions. For example, pepsin breaks down proteins in the stomach. It works best at pH 2, which means it's well suited to the acidity of the stomach. Also, increasing the substrate concentration will also increase the rate of reaction. This is because an enzyme is more likely to meet the substrate and react with it. However, if there are too many substrate molecules that all the active sites on the enzymes are full, the rate does not continue to increase. 1.10 looks at the core practical on the effect of pH on enzymes. The enzyme amylase converts starch into maltose, which is a simpler sugar. Starch can be detected as it will turn iodine solution from orange-brown to a blue-black colour. The method. Put a drop of iodine solution into every well on a spotting tile. Place a Bunsen burner on a heat-proof mat and place a tripod and gauze over the top. Put a beaker of water on the tripod and heat it until it is 35 degrees C. Try to keep the temperature constant throughout the experiment. Use a syringe to add 3 cm cubed of amylase solution and 1 cm cubed of a buffer solution with a pH 5 to the boiling tube. Using test tube holders, place the boiling tube into the beaker of water. Use a different syringe to add 3 cm cubed of starch solution to the boiling tube. Immediately mix the contents of the boiling tube, start a stopwatch and wait 5 minutes. Use continuous sampling to record how long it takes for the enzyme to break down the starch completely. To do this, use a dropping pipette to take a fresh sample from the boiling tube every 10 seconds and put a drop into one of the wells on the spotting tile. If the solution remains an orange-brown, then all the starch has been broken down, and stop the stopwatch. Repeat the experiment with different buffer solutions to see how the pH affects how long it takes for the starch to be broken down. Make sure to control the other variables, such as the concentration of the amylase solution and the temperature, to make it a fair test. (1.11) looks at calculating the rate. To work out the rate of reaction for experiments like in 1.10, use the formula 1 over the time taken. 1.12 looks at the importance of enzymes. Most of the molecules in the food we eat are too big to pass through the walls of the digestive system, so digestive enzymes break them down into smaller soluble molecules. These can pass easily through the walls of the digestive system into the bloodstream, where they can be taken into cells. Enzymes called carbohydrases convert carbohydrates into simple sugars. For example, amylase breaks down starch into maltose and other simple sugars. Carbohydrates can be synthesized by joining lots of simple sugars. For example, glycogen synthase is an enzyme that joins together lots of glucose molecules to make glycogen, which is used to store energy in animals. Proteases, such as pepsin, convert proteins into amino acids. Proteins are made by joining amino acids together, and enzymes catalyse the reactions needed to do this. A video with more information on protein synthesis will be linked in the description below. Lipases convert lipids, which are fats and oils, into glycerol and fatty acids. Lots of enzymes are involved in the synthesis of lipids from glycerol and fatty acids. (1.13b) is a core practical looking at the testing for food molecules. To test for reducing sugars, like glucose, add Benedict's reagent, which is blue, to a sample and heat it in a water bath set to 75 degrees C. If the test is positive and there are sugars present, then a coloured precipitate will form, else it will remain blue. The concentration of the sugars, the further the colour change goes. The colours go from blue to green to yellow to orange to brick red. To test for proteins, add a few drops of biuret solution. If the test is positive, the solution will turn purple. If the test is negative, the solution will remain blue. The colours are quite pale, so you will need to look very carefully at them. To test for lipids, shake the test substance with ethanol for about a minute until it dissolves, then pour the solution into water. If there are any lipids present, they will show up as a milky emulsion. If not, no emulsion will form. The more lipid there is, the more noticeable the milky colour will be. To test for starch, add a few drops of iodine solution. If starch is present, the solution will change colour from orange-brown to blue-black. If not, the solution will remain orange-brown. 1.14b looks at calorimetry. Calorimetry is the process of burning foods to work out how much energy is contained within it. A set volume of water has its temperature recorded at the start, before being heated by a burning, weighed piece of food. When the food will no longer burn, the temperature of the water is taken. The energy in the food, in joules, will equal the mass of the water, in grams, multiplied by the temperature change of the water, in degrees C, multiplied by 4.2. Then, to work out the energy in each gram of the food, so different foods can be fairly compared, divide the energy in the food in joules by the mass of the food, in grams. (1.15) looks at the transport in cells. Diffusion is defined as the net movement of particles from an area of higher concentration to an area of lower concentration, down a concentration gradient. Diffusion happens in liquids and gases as the particles are free to move, unlike in a solid. However, only very small molecules like glucose, amino acids, water and oxygen can diffuse through the cell membrane. Big molecules such as starch and proteins can't fit through. Active transport is defined as the movement of particles across a membrane against a concentration gradient, so from an area of lower concentration to an area of higher concentration, using energy released by respiration. Active transport is used in the digestive system, for example, where there is a lower concentration of nutrients in the gut than in the blood. This allows for nutrients to be taken into the blood even though the concentration gradient is the wrong way. This is essential to stop us from starving. 1.15 also looks at osmosis. Osmosis is defined as the net movement of water molecules across a partially permeable membrane from an area of higher water concentration to an area of lower water concentration. A partially permeable membrane has tiny holes, so only tiny molecules such as water can fit through, yet bigger molecules such as sucrose cannot. 1.16 is a core practical looking at osmosis in potatoes. Prepare sucrose solutions of different concentrations, ranging from pure water to a very concentrated sucrose solution. Use a cork borer to cut a potato into the same size pieces, ideally about 1 centimetre in diameter and all from the same potato. Divide the cylinders into groups of 3 and use a mass balance to measure the mass of each group, and don't forget to record them. Place one group in each solution and leave them in for at least 40 minutes. Remove the cylinders and pat dry gently with a paper towel to remove excess water so more accurate final mass is measured. Weigh each group again and record the results. 1.17 looks at calculations from the osmosis practical. To work out the percentage change in mass, use the formula: final mass minus the initial mass divided by the initial mass and then multiplied by 100. If the percentage is positive, the potato gained mass, and if the percentage was negative, it lost mass. Where the sucrose solution was less concentrated, so a higher water concentration than the cylinders gained mass as water is taken in by osmosis. Where the solution was more concentrated, so a lower water concentration than the cylinders lost mass as water was taken out by osmosis. Where the concentrations are the same, there was no change in mass. Thank you for watching. If you found this video useful, please like and subscribe, and if you have any other topics you would like me to cover, please comment down below.